everyone, my name is Carrie O'Neill. I'm a senior scientist here at the Florida Aquarium Center for Conservation. We're really excited to share a very special event with you tonight. We're here in Greenhouse 2, which is our coral archive greenhouse. And we have some of our partners here from NOAA Southeastern University. We have Dr. Joanna Figueredo and 3 of her graduate students, as well as some of our staff here from the Center for Conservation. And we're going to head over to the 3rd tray. Everybody is monitoring for spawning right now, but we actually have one of the corals is already releasing what we call bundles. So you can see in this beaker, there are some tan colored, basically little balls. Those are bundles of coral eggs and sperm. And they're coming from, oh, here's a better shot. There we go. And we use these special tripore beakers to skim the buoyant bundles off of the surface of the water. And then over time, those bundles are gonna start to break apart, releasing all the coral sperm out into the water, where we'll mix them together so that they can cross-fertilize. Joanna, can you point out which coral these are actually coming from? This one right in front of you?
Yep.
So this is one of our grooved brain coral colonies. It's currently not releasing bundles, but we're going to hope that it starts to do so again, or that we get another coral releasing bundles here in a minute. This event actually only happens once a year. This is the 10th day after the full moon of April. This coral spawns after the full moon of late April or early to late May. So this year and the past 2 years, this coral has started to spawn on exactly day 10 after the full moon. And we're not even quite sure how this coral knows that it's the 10th day after the full moon. But they do apparently have some biological ability to sense the changes in light that occur near sunset. And just their development of the eggs are ready at this time of year. And we really don't understand exactly why the corals are spawning on this particular night. But this is why we have so many people here, because we knew that the 10th day after the full moon was going to happen tonight. And luckily we have plenty of people here ready to collect the bundles. So guys, have we had more than one coral, or is it still just one? So if we only have one coral, we've got a bit of a problem because we can't make any babies. So we need a mama and a daddy. And right now, we don't want to mix the eggs and sperm from the same individual because they won't fertilize. So we're hoping to get a second colony of the grooved brain coral so that we can cross them together and make thousands of coral babies. For example, last year on a day that we had about 8 parents spawn, we were able to make 500,000 larvae, which we distributed to partners across the state of Florida. These corals in this tank are actually part of the Florida Reef Track Rescue Project. And that project is rescuing corals before they are exposed to what is called stony coral tissue loss disease. And stony coral tissue loss disease affects over 20 different species in Florida of reef-forming corals. And once the coral gets infected, most of the time it suffers complete mortality or complete death. Rachel is holding this individual so we can see its tag number. So this tag number tells us exactly what reef this coral came from, and is also linked to genetic information about the coral. Okay. So we'll have a bank of all the genetic information so that we know what parents, oh, we have another one, yay! Whoops. So this coral right in front of Jenny is now releasing bundles. So the key here, when we only have a few bundles, is that when we skim them off the surface, you actually want to make sure you don't get too much water into that beaker. Because if you only have a little bit of bundles, then the sperm can become very, very diluted if you get too much water. So if Jenny holds that beaker at a much sharper angle to the surface of the water, like more up and down, like Joanna is demonstrating, then the bundles should trickle right over the edge with very, very little water.
Maybe the point facing where you want.
Sorry, we're having technical difficulties with our camera. We're coral biologists. Our communications manager, Megan, is here helping me operate the camera. So I apologize for the shakiness and the slightly wonky camera angles. So now that we have 2 individuals, we're going to collect these bundles and we'll mix them together. It'll take a little bit of time for this bundle to break apart into eggs and sperm of the separate colonies. So I'll go back and continue on where these corals came from. So these corals were collected from the lower Florida Keys by our partners at the Florida Fish and Wildlife Conservation Commission. And there's actually 2,000 Florida rescue corals currently residing in over 18 different AZA aquariums across the country and being cared for under the Florida Reef Track Rescue Project. And the goal of this project is to preserve the genetic diversity of these species before they're lost to this coral disease that is spreading rapidly through Florida. So we're spawning the next generation of baby corals that can be used to repopulate this species on Florida's coral reef. Austin, is that from down there? Okay. So I always like to say it takes an army to save coral reefs, and we can't do any of this without our partners. So we've actually done a lot of coral spawning events with NOAA Southeastern University. We've done work together in the Keys, and now here at the Center for Conservation in Apollo Beach, Florida. And we've raised hundreds of thousands of coral embryos together. And we swap larvae, as Joanna's saying. So Joanna and her lab actually also have aquariums that induce corals to spawn in the laboratory, similar to what we do here at the Center for Conservation. So what we do is try to focus on different species, so that when she has hundreds of thousands of babies and we have hundreds of thousands of babies, we can actually just swap larvae. So after most spawning events, we'll have a larvae swap, and partners and scientists across Florida will cross their larvae so that we all kind of hedge our bets and get the greatest diversity of species and individuals that we can. So Rachel Serafin, our senior biologist here, is now recording which individuals' bundles are in that cup. This is very important as we go throughout the night to keep track of the eggs and the sperm and which coral that they came from. And another cool thing that we're going to do a little later on is actually cryopreserve the sperm from these corals. So if we ever have even greater loss of the species, using cryopreservation, these coral gametes can live indefinitely at a very low temperature and be thawed out to create new coral babies hundreds of years in the future potentially. So we're going to wrap it up for now, but continue to check back in with us on our coral spawning events. We also are expecting a lab spawning probably over this weekend, and we'll have some more spawning activity here in the greenhouse right about this time tomorrow night on night 11 after the full moon.
Thank you guys so much for tuning in. We're going to go live on our Facebook page a little bit later as the spawning picks up. So guys, definitely check back in with us in a little while. Thanks for watching.
Okay, just hit the stop.